In this video, we're going to encode the second step in our analysis workflow. So this statistical analysis step. So I'll just open up the GitLab repository for that step. Okay, so this is the statistical analysis code for the signal background and data T-trees that we're working with. So the initial step is to install the Python requirements, but we've actually already taken care of that in the Docker file when we set up our analysis image. So we don't need to do that again because they should be already in the image. Now the next step is to run this statanalysis Python script, providing the background data and skimmed signal T-trees as command line arguments. And I'll just quickly go through this Python script so you can get an idea of what it's doing. So as you can see, it's reading in the path to the background data and signal T-trees. And then it's using uproot to open up each of these T-trees and read in the MJ, so the massive leading large radius jet, into this MJ sig or MJ data or MJ background variable. And then it's plotting histograms of the either the background plus signal, the background only, or the signal only together on one canvas. And then it's also plotting the data histogram with error bars. And then lastly, it's calculating the chi-squared and associated p-value for the comparison between the background plus signal and the data. And then it's printing out this chi-squared and p-value onto the canvas and saving the resulting figure. And then it's also saving the p-value that we calculated for this chi-squared statistic into this p-value recast.txt. So this is meant to be sort of the result of our very simplified analysis workflow. Okay, so let's open up the specs.yaml file, or steps.yaml, sorry, and encode this second step. So we'll start by just copying the event selection because there's going to be a fair bit of repetition. So I'll first rename it to statanalysis. And then we're going to need to update the image that the step runs in. So we can do that right off the bat. And the place we find that, remember, is in the container registry. So we go into the image and select the latest tag. So that's this one that got produced 2 hours ago. Okay, so we can copy that in, and remember the image tag has to get specified separately. Okay, and then the other thing that we're going to need to update is, of course, the bash commands that actually get run inside of this Docker container. So again, we are going to need to do this Kerberos setup because we're going to be reading in the background and data T-trees from EOS that already have the skimming pre-applied. We don't need to source the release setup because we're not in an Atlas software container anymore. We're in root conda base image. And then rather than copying in the signal file with xrootd, we're instead going to be copying in the background T-tree. So we'll call it BKG file. and we'll save it to background.root. And then we'll also copy in the data file. And save it to data.root. Okay, so now we can run our Python script. So we'll replace the event selection command with the command to run our Python script from the README. And the only thing that's going to need to be updated here is that rather than reading in the skim signal from the image, we're going to read it in as a variable because it's being supplied from the previous step in the analysis workflow. So we'll call it skim signal. And then the last thing that we want to do is output the files that get produced by our workflow to an output directory so that it can be published and made available as the output of our recast workflow. So to do that, I'm going to make an output directory, call it output here. And I'm going to see what gets produced by the analysis workflow by going back to the README. So it says the script will produce two output files. It produces histos.png, so that's these stacked histograms of signal and background overlaid with data that get produced by the script. And then also this p-value recast.txt that just contains the p-value for the chi-squared comparison between signal plus background and data. And there's also an example here of what the histos.png could look like. So there's a standard model background plus signal shown in red here, and standard model background only in blue. And then that's overlaid with the actual data, which in this case was generated to be consistent with the standard model background. So we're not expecting to see an excess, unfortunately. And then there's also, it also plots the signal only as a dashed histogram, so you can see what the shape of the signal looks like. Okay, so these are the products of our recast workflow. So we'll copy both of these out into the output directory. And then the last thing that we need to do is update the variable that gets published by this step. So that's going to be the output directory that we're writing our output files to. So I'll replace output file here with output dir, and then we'll call it something a bit more meaningful like result dir. So that should be everything that we need for this second step of our analysis workflow. And then the next thing that we can do is go into our recast.yaml file and add a test for the second step to make sure that everything's running as expected. So again, we'll copy the test from the event selection and just make some modifications. So I'll add a test stat analysis, and we're gonna have to update which step is actually being tested. So now it's the stat analysis step in this steps.yaml file. And then we're also gonna have to update the parameters that get supplied. So we're now reading in a background file, this BKG file. Oops. And if we look at the directory where our recast inputs is stored, we see that the background file is called background.root and data is data.root. So we can replace that here. Okay, and then looking at our steps.yaml file, we also have to specify the data file and also where to find the skimmed signal and where to where the output directory is going to be located. So let's add those in. I'll first add the data file because that's also going to get read in from EOS. Okay, and then the output dir is going to be output to the work dir, and we can call it maybe recast results. But the last thing that we need to do is supply the skimmed signal T-tree that got produced in our previous step. And it's not actually clear at this point how we can do that, because remember, the output from that first test got produced in this unique, I'll just do an ls, this unique recast -test dot a bunch of numbers and letters directory. So it's not obvious how we would read it in from there. So what we're going to do is we're going to set up a common directory that both of these tests have access to. And so if we go over to the recast docs, there's an explanation of how to do this under, where is it? Using test data. So what we have to do is add this data field that specifies a directory called test data where the inputs and outputs are going to be stored. And the way that that's specified is with this read dir variable. So by specifying test data as this data field, what this is doing is setting this read dir 0 variable to the path to this test data directory. So we're actually going to have to update that in both of the steps. So I'll just copy and paste that in. And then we can just update work dir to read dir 0, so that now the test event selection is going to be outputting the signal skimmed into this test data directory. And similarly, the statistical analysis test will be outputting the directory that it produces to this test data directory. And now we can also read in the skimmed signal file from this read dir. I'll add that in here. And it'll be the exact same as what got output in the previous step. Okay, so let's try that out and see how it goes. Now we're going to have to rerun this first event selection test so that it produces the skimmed signal T-tree in this test data common directory. And then we can try testing our statistical analysis step.